Welcome to this lecture on the fundamentals of genetics. Today, we are diving into a truly fascinating topic called the dosage compensation mechanism. If you have ever wondered how nature ensures fairness and balance at a microscopic level, especially between males and females who have different sex chromosomes, you are in the right place. This mechanism is absolutely essential for life as we know it. Let us begin our journey to understand how cells solve this critical genetic puzzle. To understand our topic, let us start with a simple analogy. Imagine you are managing two identical shoe factories. The first factory has two identical machines producing shoes, while the second factory only has one machine. If all machines run at the exact same speed, the first factory will produce twice as many shoes, creating a massive imbalance in your inventory. To fix this, you have a few options. You could turn off one machine in the first factory, or you could make the single machine in the second factory work twice as fast. Keep this idea in mind because this is exactly how cells handle genetic imbalances. Now that we have our factory analogy, let us look at the actual biological problem. In many species, including humans, females possess two X chromosomes, while males possess only one X and one Y chromosome. Because the Y chromosome is very small and contains very few genes, males essentially have only one copy of most X-linked genes. Without a regulatory mechanism, females would transcribe twice as much RNA from their X chromosomes as males do. This inherent genetic inequality is solved by dosage compensation. It is the genetic regulatory mechanism that adjusts transcription rates so that the final amount of gene product is perfectly equal in both males and females. Understanding what this mechanism is leads us to ask why it is so critical. A strict balance of proteins is absolutely required for normal cellular function and development. If a female were to produce twice the amount of X-linked proteins as a male, the delicate stoichiometry of protein complexes in the cell would be completely shattered. This kind of genetic imbalance is usually lethal to a developing embryo. In the real world, we can actually see the visible effects of this balancing act in the beautiful coat patterns of calico and tortoiseshell cats, where random inactivation of X chromosomes creates their unique, patchy fur. It also helps doctors understand various genetic disorders. Before we look at specific animals, let us understand the math behind how cells balance their output. We can represent this with a simple mathematical model. The formula is E equals N times R. Conceptually, this means the total amount of product a cell makes depends on how many copies of the gene it has and how fast those copies are working. In this equation, E represents the total expression level of an X-linked gene, measured in the amount of protein or RNA produced. The variable N represents the number of active X chromosome alleles present in the cell. Finally, R represents the transcription rate, which is how fast each active allele is being read. If the number of active alleles increases, the transcription rate must decrease to keep the total expression constant. Now that we have our formula, let us see a quick conceptual example of how it maintains balance. Suppose a female has two active alleles, so N equals 2. Operating at a base rate, R. Her total expression is 2 times R. Now, consider a male who only has one allele, meaning N equals 1. To achieve that exact same total expression E, the male's single allele cannot just work at the normal rate. It must work twice as hard. Therefore, his cells double the transcription rate to 2 times R. This perfectly matches the female's total expression, showing exactly how the math translates into biological reality. This mathematical balancing act brings us to how mammals, including humans, actually perform this in real life. Mammals use a process called X inactivation. Remember our shoe factory where we turned off one machine? That is exactly what happens here. In female mammals, one of the two X chromosomes is tightly condensed and permanently silenced. This dense, inactive structure is called a bar body. The golden rule for mammalian dosage compensation is that a cell will inactivate all X chromosomes except for one. This ensures that only one X chromosome remains active and producing proteins, regardless of how many X chromosomes are originally present in the cell. Knowing that mammals shut down extra chromosomes, we can calculate exactly how many bar bodies will form using a simple formula. The formula is B equals N minus 1. Conceptually, we're just taking the total number of X chromosomes and leaving exactly one turned on. In this equation, B represents the total number of bar bodies, which are the silenced X chromosomes. The variable N represents the total number of X chromosomes present in the individual's genome. The number 1 represents the single X chromosome that must remain active for the cell to function properly. If the total number of X chromosomes increases, the number of bar bodies will also increase to maintain that strict balance of one active chromosome. Let us apply this formula to typical human genetics. We want to find the number of bar bodies in a typical female and a typical male. For a typical female with 2 X chromosomes, our given value for N is 2. We substitute this into our formula, giving B equals 2 minus 1. This results in 1 bar body. For a typical male with 1 X and 1 Y chromosome, the total number of X chromosomes is just 1. So N equals 1. Using the formula gives B equals 1 minus 1, which equals 0. This means the male's single X chromosome remains fully active and no bar bodies are formed. This perfectly equalizes the active X chromosomes between the sexes. Now that we have seen typical cases, let us look at an abnormal genetic condition. Consider a patient with Klinefelter syndrome, who has 2 X chromosomes and 1 Y chromosome. We need to find out how the body compensates for this. First, we identify our given information. The patient has 2 X chromosomes, so N equals 2. We choose our formula, B equals N minus 1, because the human body will still attempt to maintain dosage compensation. Substituting N equals 2 into the formula gives B equals 2 minus 1. The final answer is 1 bar body. Physically, this tells us that even in abnormal conditions, the cell silences the extra X chromosome to prevent a toxic overdose of X-linked proteins. While humans shut down an extra chromosome, other organisms take a different path. Let us look at the fruit fly, Drosophila melanogaster. They use a completely different strategy to achieve the exact same goal of genetic balance. Remember our shoe factory where we sped up the single machine? That is what happens here. In fruit flies, the female keeps both of her X chromosomes fully active. To keep up, the male hypertranscribes his single X chromosome. Instead of turning things off, the male's single genetic machine simply works twice as fast to match the female's total production. To understand exactly how hard the male fruit fly's chromosome must work, we can rearrange our earlier gene expression formula. The formula is R male equals E female divided by N male. Conceptually, this demonstrates that the male must divide the total required expression by his available number of chromosomes. In this equation, R male represents the transcription rate required by the male's single X chromosome. E female represents the total target expression level set by the female, which is the goal the male must reach. Finally, N male represents the number of X chromosomes in the male. If the target expression E female increases, the male's transcription rate must also increase proportionally to keep up. Let us put this formula into practice with a concrete example. Assume a female fruit fly has a total expression level of 100 units for a specific gene, with her two alleles working at a base rate of 50 units each. We need to find the transcription rate for the male's single X chromosome. We know the target expression, E female, is 100, and the male has exactly one X chromosome, so N male is 1. We substitute these values into our formula, giving R male equals 100 divided by 1. Solving this simple division yields 100 units per allele. Physically, this means the male fruit fly achieves dosage compensation by doubling the output of his single chromosome to perfectly match the female. Now that we have calculated the rates, let us look at this diagram summarizing both strategies. On the left, we see the mammalian approach. Notice the female cell has one active X and one condensed bar body, while the male has one active X and a small Y. Both arrows lead to identical protein pool boxes, showing that silencing an extra chromosome creates balance. On the right, we see the Drosophila approach. The female has two active X chromosomes, but look at the male cell. His single X chromosome is enlarged with a bold arrow labeled hypertranscription. Despite these completely opposite methods, turning a machine off versus speeding a machine up, both strategies successfully result in the exact same amount of protein, ensuring the survival of the organism. We have covered a lot of ground today, so let us review our key takeaways. First, dosage compensation is a vital evolutionary mechanism that ensures males and females produce the exact same amount of X-linked gene products. Second, we learned that without this system, the resulting genetic imbalance would cause severe developmental defects, or even death. Finally, we saw how nature solves this mathematical problem in beautifully diverse ways. Mammals silence an entire extra chromosome, while fruit flies double the transcription rate of a single one. You now understand how cells perfectly balance their genetic output to sustain life. Great job!